Bacteria, they're everywhere, and they're microscopic. These tiny organisms are so small that millions of them can fit on the head of a pin. These tiny organisms play a big role in our world. They can be found in soil, water, and even in extreme environments like hot springs and deep sea vents. Some bacteria are helpful, like the ones in our gut that help us digest food. These beneficial bacteria are essential for our health and well-being. Other bacteria can make us sick. Harmful bacteria can cause infections and diseases ranging from mild to life-threatening. To understand bacteria better, scientists use a technique called Gram staining. This method helps them identify and classify bacteria. Hans Christian Gram, a Danish scientist, developed Gram staining in 1884. His discovery revolutionized microbiology. It's a simple test that tells us a lot about bacteria. By staining the bacterial cells, scientists can observe their characteristics under a microscope. By looking at the results of this staining process, we can group bacteria into two categories: gram-positive and gram-negative. This distinction is based on the structure of their cell walls. This classification helps us understand how to treat bacterial infections. Different antibiotics are effective against different types of bacteria. Imagine you have a mystery bag of Skittles and M&Ms. You want to separate them, but you can't see inside the bag. You want to separate them, but you can't see inside the bag. It's a tricky situation. Gram staining is like having a special filter that separates the Skittles from the M&Ms based on a unique property of their candy shell. This filter makes it easy to identify and classify them. Gram staining involves a series of meticulous steps that are crucial for differentiating bacterial species. First, we take a smear of bacteria and heat fix it to a slide. This process ensures that the bacteria adhere firmly to the slide. This is like sticking our Skittles and M&Ms to a piece of tape, ensuring they don't move around. Then we add a purple dye called crystal violet, which is the primary stain in this procedure. Both types of bacteria will absorb this dye and turn purple, making them easier to see. Next, we add iodine, which acts as a mordant, helping the dye stick better to the bacterial cell walls. Think of iodine as a glue that makes the dye stick to our candy, ensuring it stays in place. Then we wash the slide with alcohol, which is a critical step in the process. This is the critical step where the magic happens. The alcohol decolorizes the slide, affecting different bacteria in different ways. Gram-positive bacteria hold on to the purple dye tightly even after the alcohol wash. They remain purple and are easily identifiable. Gram-negative bacteria, on the other hand, lose the purple dye and become colorless, making them harder to see initially. Finally, we add a pink counterstain called safranin, which provides a contrasting color. This stains the colorless gram-negative bacteria pink, making them visible under a microscope and completing the gram staining process. Gram-positive bacteria have a thick cell wall made of peptidoglycan. Think of it like a thick, sturdy brick wall. This thick wall traps the crystal violet iodine complex, preventing it from escaping during the alcohol wash. Some common examples of gram-positive bacteria include which can cause skin infections and which can cause strep throat. These bacteria are like those pesky weeds with deep roots that are hard to get rid of. Gram-negative bacteria have a more complex cell wall structure compared to gram-positive bacteria. They have a thin peptidoglycan layer sandwiched between an inner and an outer membrane, which is unique to their classification. Imagine a thin sheet of drywall between two layers of plastic wrap. This analogy helps visualize the protective layers. This structure makes it easier for the crystal violet iodine complex to escape during the alcohol wash, leading to a pink or red appearance under the microscope. Examples of gram-negative bacteria include Escherichia coli, found in the intestines, and Salmonella, which can cause food poisoning. Treating infections caused by gram-negative bacteria can be trickier because their outer membrane acts like a shield, preventing many antibiotics from entering. This makes them more resistant to certain antibiotics, posing a significant challenge in medical treatments. Gram staining is a fundamental tool in microbiology, essential for identifying bacterial species. It helps us quickly identify and classify bacteria, guiding treatment decisions and improving human health by enabling targeted therapies. So next time you hear about bacteria, remember the power of gram staining and the fascinating world of these tiny organisms, which play crucial roles in both health and disease.